Okay, so for the pattern inheritance here, what we're going to study in this case, for today, we're going to explain the term, what is meant by first filial generations, or called F1, and what's meant by second filial generations, F2. And we do need to look at another term called parental generation, or sometimes we call it a P generation, parents, okay? So F1 and F2. And for today, we are going to use a genetic diagrams. Okay, we have to learn how to use genetic diagram or the Punnett square. Okay, we will learn about this Punnett square. To solve the problem includes, for today, we're going to look at the monohybrid and dihybrid crosses. Okay, and then the next lessons, we're going to look at, okay, adults modification involve autosomal linkage, sex linkage, codominance, multiple alleles, and also the gene interactions. We're going to look at those. Okay, the ones we're going to learn the next lessons. Okay, and then followed by the term what it means by the test cross. What we need to use a test cross, and we're going to use genetic diagram to solve the problem involving the test cross. Okay, so the basic principle of the hereditary, in this case, you guys can go through it, very simple, because when Mendel was the one, Mendel was the one that actually carried out this experiment through his observations onto this garden pea. Then he came out with those ideas, how the process actually do take place, which what actually determines the characteristics of the offspring. Okay, so he observed these seven. He observed these 7 characteristics, where we are going to focus later in our syllabus. We're going to focus in terms of seed color. So now you have to know that those bold means dominant trait. Now be careful what is in dominant trait. When you use the word dominant trait, trait means characteristics, so means that refer to phenotype, not allele. It's a dominant trait. Are you clear? And we do have the recessive trait. Trait again means that phenotypes, not genotypes. Okay, so we're going to focus in the seed color, where we're going to compare between yellow and green, and then also the shape of the seed, either smooth or round. Sometimes call it round, and versus wrinkled. Okay, so we have these 2 as our model to study monohybrid and dihybrid crosses. Okay, I will tell you what it means by the monohybrids and also the dihybrids. Okay, so Mendel's always started his experiment by crossing the plant from 2 different, highlights the word true breeding or pure breedings. So when you answer the question, these 2 words actually is very, very important. If you do not understand these 2 words, pure breedings or true breeding, you will not be able to continue to answer the question. So what is mean true breeding and pure breeding? Same meaning, both of them means that they are in their homozygous conditions. True breeding and pure breeding means that they are homozygous. So sometimes question will ask you, okay, please draw a genetic diagram to show how two pure breeding yellow plants can result in the formations of blah, blah, blah. So if you do not know what is meant by pure breedings of true breeding, then you do not know what is their genotype. So basically pure breeding and true breedings means they are homozygous. So this one, Mendel actually labeled them as the parental generations or P generations. The offspring will form the first filial generations, or we call F1 generation. So the cross, so basically we started with the P, parental and parental. So their offsprings is called F1. So when F1 cross with F1, then we get the F2. Okay, filial 2, or the offspring of the filial 1. Okay. So taking the seed as example, later we're going to look at it. Mendel showed that when a true breeding yellow pea and true breeding green pea, when they cross together, they always get the F1 produce the yellow seed. Later we have to show how we can get this result. Okay. And the second phyllodes or the F2 generations result from the cross between F1 individual always give us, again, a ratio of 1 green to 3 yellow. So later we will be doing this, okay, to show how we can get this, okay, result. So Mendel used a term recessive and dominant to describe the trait. Okay, so we have recessive trait and dominant traits. Dominant trait means that appear more. Okay, so yellow color dominant trait. Okay, in F2 generation. Okay, so Mendel actually come out with 2 laws. Mendel come out with 2 laws. The first law actually is the law of the segregation. Law of segregation. So basically means that they refer to the meiosis, because we know that in the meiosis, what we're going to do here is we are going to segregate the homologous chromosome. So means that if you have the dominant allele, okay, chromosome, homologous chromosome, you have dominant allele big A, recessive allele small a. After the ends of the meiosis, you're going to segregate them into 2 gametes. So means that each gamete only will have 1 allele. So either dominant or recessive. So this is Mendel's law, first law. Law of the segregation. Okay. Mendel also come up with the second law, which deal with the independent assortments. Okay, Mendel come out with a second law, law of independent assortments. Are you clear? So law of independent assortments basically go back to our independent assortments that we have learned during the meiosis, where we're going to make, because if you look at the law of independent assortments, involve 2 chromosomes. So look at this. So I'll draw it here. Okay, hopefully I can. So if the gene located at 2 different chromosomes, so I can have the allele big A, allele small a, allele big B, allele small b. So in this case, we will get the genotypes of the gametes with big A, big B, small a, small b. But because of independent assortments that we have learned just now and on the last lesson, it may be this way. When arranged, it might have the big A, small b, big B. So at the end, we're going to have another 2 different combinations where I'm going to have the big A, small b together, small a, big B together, and all of them equal chance. Can I see that? So means that this is due to what? This is due to the law of the independent assortments, a law of independent assortment. So law of segregations, later we're going to use it for what? For monohybrid. Means that only 1 gene. We look at 1 gene. Law of independent assortments, we're going to look at 2 genes. But these 2 genes located on different chromosomes. Okay, because the next lessons, we're going to look at 2 genes located on the same chromosome. Okay, so today we're going to look at these 2, law of segregations and also law of independent assortments. Okay, so this one you can go through it. So in this very important few things, highlights number 17. Why? Because when you choose a symbol in the exam, when you choose a symbol, you must use capital letter for dominant, small letter for recessive, unless they have more than 2 alleles. Let's say dominant have 2 or 3 alleles, then we have no choice. We cannot use capital letter and small letter. Okay, so if more than 1 allele, what we're going to more than 2 allele, normally we're going to put it like this. For example, blood group IA, IB, IO. So we will use this form if the allele more than 2. Okay, clear? So in this study, in this case, we're going to use this big Y as a dominant allele code for yellow color seeds. I cannot use G because the symbol must be the same. So must we use small y for the recessive allele, code for green. Big R, dominant allele, code for round. Small r is the recessive allele, code for wrinkled seed. Okay, so we're going to start it with the monohybrid cross. This thing you have learned it before, just that in CIE, there are some rules that we have to follow. Okay, so what are the rules? First thing, you have to follow this, guys. You have to follow this. So let us start this one first, yeah. Very simple, actually very simple. You just need to follow. Okay, later I will highlight what you need to put in. So we look at this law of segregation in monohybrid cross, right? In monohybrid cross. So I have parent 1. So parent 1 produce yellow seed. Parent 2 produce green seed. And they are, because we always say that they are true breeding. True breeding basically means that homozygous. So we're going to start with the parental phenotype, yellow seed, parental phenotype, green seed. Okay, so yellow seed, they are homozygous. So parental genotype, big, right? Big Y. Parental genotype will be small y for green. And because of law of segregations, look at this. Okay, so we have two. Can I see that? We have 2 chromosomes, right? 2 chromosomes, each one carry big Y allele. So when you segregate, so all the gametes will have big Y for parent 1. Parent 2 will have small y. So here I want you guys to highlight first thing. You must include probability. Not nice color. Let me see what color I can choose better. Okay, so highlight here. This is the must. You must put in probability. Are you clear? You must put in probability. And then you can draw your Punnett square. So we have parent 1. So all big Y allele. Parent 2, all small y allele. So can you see that? Another one thing is gamete must be circled, yeah, guys. Gamete genotype. You must circle, must be circled. Are you clear? You must circle the gamete genotypes. If no, consider wrong. Okay. So then in your Punnett square, you need to have the 3 things. You must show the genotype. You must write down the phenotype. I will tell you guys later more. Okay? And also the probability. This is the must. Okay. Okay, so I summarize here few things that you need to know. So first, you need to determine what is the parental phenotype. So very simple, how to check the parental phenotype. How we know parental phenotype? So from question. Question will always tell you, right? From the question, you always know what is the parental phenotype. Then next, after you know the parental phenotypes, then you have to figure out what is the parental genotype. So this one you have to figure out. Okay, either big Y, big Y, big Y, small y, or small y, small y. So which one you have to figure out? If you can't figure out, you won't be able to continue. So again, how you know? From the question. Then step number 3. You need to come out with the gamete genotype. Okay, so these are the important things that you need to write. Importance. First, you must have the probability of the gamete genotype. And you must circle the genotype, gamete genotype. Okay, then number 4. You can construct the genetic diagram. So inside genetic diagram, 3 informations that you need to put in. First information, you need to have the genotype. Number 2, you must have the corresponding phenotype. And number 3, you need to have the probability for each. Okay. And then you will be able, from here, you will be able to determine the expected ratio. So these are the 5 steps that always involve. Okay, this is, I say that if you want to play safe, this is the answer, because sometimes question may not require you guys to put in probability. Okay, but the question sometimes they need, then you don't have, then you lose the mark. Okay, so this is the complete set how you answer the question. Always remember, for gamete genotypes, you must have the probability of the gamete genotypes, and you have to circle the gamete genotype as well. Okay, and then for the genetic diagrams, you need to have the genotype, phenotypes, as well as the probability. Okay, but sometimes for phenotypes, I can use, okay, for phenotypes, I can use the key, okay, or symbol to represent, because I don't want to write so many. Okay, I will show you guys later. Okay, so use back the same thing, then we continue. So first, because we know that this is a F1. Okay, this is a F1, right? This is a parent, so this one is the F1. So if I cross the F1 with F1, so Mendel then allowed the F1 individual to self-pollinate to yield the F2 generations. So draw a genetic diagram to show the probable results of the self-pollinations between F1 generations. Because I know here, F1 is the yellow seed. So can I see that? F1 phenotypes, yellow seed. What is the genotype? Because I know that F1 is the outcome from the cross of the parental, pure breeding parental, so it must be heterozygous. So can I see that one big Y, one small y? So when come to the gamete genotypes, always remember, first thing, probability. Second thing, very important, please circle. You must circle your gamete genotype. So once you circle your gamete genotypes already, then you have this, what we call the genetic diagram. So it means that now I will have first F1 generations, second F1. So big Y, small y, big Y, small y. So when I combine big Y, so big Y, big Y. And then big Y, small y, big Y, small y, small y, small y. So what you're going to do here is you multiply the probability. So half and half. So you get 1/4 each of them. So when you guys need to get the phenotypic ratio, the correct step here, you need to show, you need to show the ratio. Okay, how you show the ratio? So using probability. So yellow seed, we have how many times? 3 times. Each of them is 1/4. Green seed, 1 times, 1/4, so 3 to 1. Very important, you have to show that yellow seed is 3, green seed is 1. Don't simply tell me 3 to 1. Because you tell me 3 to 1, I do not know 3 represent. I do not know 3 represents green or 3 represents yellow. Okay, so again, this is the whole process. I want to summarize again that very, very important. Parental phenotypes, you always can get it from a question, because questions will be the ones that tell you what we want to cross. And then from the question, you'll be able to get the parental genotypes. And then from parental genotype, you'll be able to get the gamete genotype. So from here, you will be able to find out what is the probability of the gamete genotypes. And very important, you need to circle the gamete genotype. Then you proceed to the genetic diagram. So inside genetic diagram, you must have these 3 items, genotypes of each box, and then phenotypes, corresponding phenotypes, sorry, and then with the probability of each phenotype. From there, you will be able to get the ratio. The ratio always have to be calculated from the probability. Okay. So this is already done for monohybrids, very simple. Let us look at the dihybrid cross. So again, for dihybrids, then you involve the law of independent assortments in dihybrid cross. Okay, law of independent assortments. So when a true breeding, guys, again, we use the word true breeding, so it must be homozygous. Yellow round seed cross it with the green wrinkled. Okay, parent 1 and parent 2. So from here, you can see that because question already tell you, right? Yellow seed, yellow round and green wrinkled. So parental phenotype is given. So what is the parental genotype? Because from the question, true breeding, right? So true breeding, it must be homozygous. Can I see that? Big Y, big Y, big R, small y, small y, small r, small r. Okay? So what is the gametes? So in this case, no matter how you arrange them, okay, see here, you have big Y, big Y, big R, big R. No matter independent assortment take place also, you won't see the effect. You're going to get big I, big R as my gametes. So very important, just now I said that you must have the probability and circle. Probability and circle your gamete genotype. So when you cross it, then you can see that I will get the heterozygous. Always remember when you cross two different variety or two different traits, when you cross them, pure breeding, I always will get the heterozygous. So that's why you can see that what I have here, I will have the probability, I have the phenotype, I have the genotypes in my genetic diagrams. And I do know that F1 generations must be heterozygous. Okay. So when now then allow F1 again, so okay, F1 individual to self-pollinate, so means that the same F1 with F1, I will get F2 generation. So because from the question, I know that all my F1 will be yellow seeds, yellow round seed, yellow round seeds. But from the question, I know that they are F1. F1 must be heterozygous. So now here, independent assortments take place. Why independent assortments? Now look at this. I have big Y, small y, big R, small r. If I arrange this way, then I can pack big Y, big R together. I can pack small y, small r together. But not necessarily big Y, small y. I can arrange small r, big R due to the independent assortments, correct or not? So it means that I may have the ability, I may have the chance to get big Y, small r together, small y, big R together, and each of them have equal chance, so 1/4. So always remember, can you see that? I will always put in my probability. Don't forget. Okay, if question don't need it, then leave it lah. But if a question need it, then you gain the marks. Okay, so don't forget, circle your gamete genotypes. Okay, circle gamete genotype. Then you put into this Punnett square. Okay, put in Punnett square. So you can see that in my Punnett square, you always will have the genotype, phenotype, and also the probability. Now, what I say here is, if you don't want to, guys, if you do not want to write every time, okay, let's say I don't like to write, oh, yellow round so many times. So I can put a symbol. Okay, yellow round. I put, okay, with shading. Okay, yellow round, I put shading. Yellow wrinkled, let's say wrinkled, maybe. Okay, with shading. Okay, green round, no shading. Green wrinkled, yeah, no shading. So I can put a key for my symbol. You can write. There's another way to do it. If you do not want to write yellow round, yellow wrinkled, and write so many times. So this is what you can do. Okay, put a key outside. Okay, so what is the F2 phenotypic ratio? So phenotypic ratio here, you can see that I have 9 boxes. 1, 2, 3, 4, 5, 6, 7, 8, 9. Can I see that? 1, 2, 3, 4, 5, 6, 7, 8, 9 appear 9 times. Each of them is 1 over 6. So 9 multiplied 1 over 6. Yellow wrinkled, green round, 3 of them, each of them 3, and 1 over 6. And last one, green wrinkled, 1 over 16. Okay, sorry, 1 over 16. So get the ratio 9, 3, 3, 1. So this is a typical ratio when you cross heterozygous with heterozygous. Are you clear? But again, don't tell me what, if I ask you, what is the phenotypic ratio? Don't give me 9, 3, 3, 1, because if you give me 9, 3, 3, 1, I will ask 9 refer to who? 3 refer to who? Unless your phenotypic ratio 1 to 1 to 1 to 1, 1, 1, then okay lah, because I know. Okay, but if a 9, 3, 3, 1, you have to mention which one. Yellow round, yellow wrinkled, green round, green wrinkled. Okay. So one thing I need to specify here is when you do the independent assortments, when you assume the independent assortment take place, means that now that 2 genes, in this case, you have the gene that code for the color, gene code for the color, gene code for the shape. So 2 genes. Are you clear? 2 genes involved. So these 2 genes must be located on, okay, must be located on different chromosomes. Okay, so let me show you what this means. When you talk about you want to have the independent assortments, very important, guys. When you talk about independent assortment to take place, we already assume that these 2 genes will be located on 2 separate chromosomes. So it means that, let's say, big Y, small y, big R, small r. Can you see that? So it means that these 2 genes, they are not on the same chromosome. They must not be. Then only you can use the independent assortments. Okay, so it means that I can have this possibility or another possibility here. So what are the possibility? I can have a big Y, small y, and then small r, big R. Can I control? I can't control. I cannot control how they align. So it means that if you separate them, you will have big Y, small r, small y, big R, big Y, big R, small y, small r. Due to independent assortments, so each of them have equal chance. So it means that 1 over 4, 1 over 4, 1 over 4, 1 over 4. Are you clear? So this is what we call the independent assortments. When you deal with independent assortments, you already assume that these 2 genes, if question didn't say anything, means that these 2 genes, we're going to assume they are located on 2 different chromosomes. They are not on the same chromosome, then only can use independent assortments. If they are located on the same chromosome, which we're going to learn in the next lesson, if they are located on the same chromosome, what will happen? Look at this, big Y, let's say big R, small y, small r. So means that you only will have 2 possible gamete genotypes, big Y, big R, small y, small r. That's all. Big Y cannot be small r, small y cannot be big R. Why? Because they're located on the same chromosome. When they're located on the same chromosome, there is no independent assortment. So when there are no independent assortments, then we only have 2 possible gamete genotype, big Y, big R, small y, small r. So this one we're going to learn its next lesson. Okay, so with this, I have done for today's class.